with the administrative stuff. Hi, everyone. Oh my God, this is too high. Is this better? There's an echo. Yes. Yeah, maybe. Hi. It's still an echo. No. Yes. No. No, I think it's fine like this. Okay. So welcome for the second day. A few administrative things before starting. Tonight, we're gonna have a party at Zumai, starting from 7 p.m., but you can come whenever you want. And please register via this link so we know how many people will be there around. We reserved a part of the bar. And it's a super cool one near Castellan. So yeah, all of you are welcome. Yeah, you can find this QR code also around the room. There is an echo, yeah. But do we care? Yeah, okay, perfect. Yes. Thanks to our technical expert. So the program, this is the program for today. And just a reminder again, you can't bring any food here, any drink here, okay? Only in the areas that are, Maya, there is no battery, only there, sorry. Yeah. You have to take the whole backpack. And exactly, very important, we want to recycle the plastic of the badges. So please, I mean, not recycle the plastic per se, but all the badges. So please return your badges after the conference. You can give it to us there on the table or put it in the boxes that are there. And I think that's it. Okay, so I'm gonna introduce Dr. Rochelle Ackerley for the first talk of the day. And yeah, so a few things about Rochelle. So Rochelle received her PhD in physiology at the University of Bristol. And then she worked as a postdoc in the UK and Sweden. There is still the eco, I think. And so I'm gonna try to do like this. She's now group leader of the Somatosense Lab in the Laboratoire des Neurosciences Cognitives at Aix-Marseille Université. And she also has recently been awarded with the bronze medal of the CNRS. And since 2020, she's the president of the International Association for the Study of Affective Touch. Rochelle research focuses on understanding the mechanism of perception and particularly of affective touch. And yesterday she was with us for the Cine Débat. We already had some very interesting questions coming up. And so thanks again, Rochelle, for participating yesterday. And today we can continue the discussion with a super interesting talk on the scientific mechanism underpinning the affective touch. So thank you, Rochelle, welcome, and the floor is yours.
Merci. Hello, and thank you all for being here. It's a pleasure to be here in front of all of you, and thank you to the organizing committee because as in previous years, I've always enjoyed the Neurodays Symposium. It's very interesting to hear from you as well. So today I'm going to talk to you about society and neuroscience, but the world of touch. So my research is focused very much on touch, but it's also exploratory touch and how touch makes you feel. So today I'm going to tell you about some of the things that I do in my research. So I really like this quote here because it really does explain what touch is. It is a language that we understand instinctively. We don't need to learn to touch. It's innate. It's the first sense that we have when we're born. Our skin is something that covers our body. It's always there. And we just know touch. We don't have to learn it specifically off our parents or anything like that. And it's a language which is universal. Of course, touch is different in different countries, but it's something that's common to us all. And so when we think about touch, we typically think about when we go out and we actively touch something. So for example, here, and there's a duration to the touch. It's either rough or smooth. It has edges. There is pressure. This is, when I say touch, this is what most people think of. However, for me, there is much, much more to touch than this. And this is why I want to explore it today with you all. So when we kind of explore other things in touch, touch also has these different senses here, such as wetness or softness, and it's got temperature. It can be sticky or have a texture. So there's already a level of complexity here in the touch. And these are some things that I'm investigating myself, and we know very little about how this is encoded. Then we go into kind of a more deeper level of touch. And so touch can be pleasant, it can be soothing, it can be comfortable. So this is more affective touch. And this is positive, this is nice, this is what I research. I like researching positive, affective touch. However, there's also a more unpleasant side to touch. It may not necessarily hurt, but touch can certainly be unpleasant. When you touch something which is very sticky or you think it's dirty, you don't like it, and you know very quickly that you don't like it. It can go all the way through to painful touch. And so we've got, for example, the softest pressure here, all the way through to something which has really hurt you. So for me, touch is a very, very big domain. Oop, this one. So how is touch actually encoded then in our skin? Well, we have hundreds of thousands of receptors in our skin. And the receptors here, the A beta mechanoreceptors, which signal what we think is more discriminative touch, they send the information very, very fast to the brain. We also have thermoreceptors, which are A delta, which send the information quite fast, but we also have C fibers here, which start to send the information a lot more slowly. And so then we also have nociceptors, which is C fibers, and we have something special here, which is I'm going to talk to you more about today, C tactile mechanoreceptors. And these are C fibers again, and they send the information relatively slowly to the brain. And so there's a distinction between the information which is coming in very, very fast and more kind of slowly. So this is more of a discrimination here of your touch, and this gives more the effect. Basically, I like to kind of divide it into, you've got in touch, you have touch itself, but you also always have temperature, and you may also have pain. So it's already a complex system here. So how do you actually feel? How do you feel? Like I said, you have more the discriminative side for exploring your world. And then this may go into more kind of complex things such as wetness and texture. And then we've got this more affective side. We have pain and pleasantness. But I just want to add that there's also the side of touch that it can go wrong. And so what do we do when it goes wrong? It may be somebody has got diabetic neuropathy and they may lose some of their sense of touch, or like this person here, they've actually had an amputated arm. How can we restore the sense of touch? How can we apply the sense of touch? So this is also something which is very interesting to investigate. So there is this kind of divide between these discriminative and affective sides, and people typically do divide this because you have to do it to actually work on it. And so you've got here examples of what we call discriminative touch, and here more affective touch, which is more the society side. However, for me, touch is always nearly both of these things. When you pick up an object, you have this discriminative side, but you also do have some appreciation of the touch. So whether it's a pleasant or unpleasant, something like that. And vice versa, when you have a nice hug, it may feel pleasant, but you actually have the duration of the hug. So this is very much intermingled, and we have to think about both of these things when we think about touch. So when we actually look at the skin, what do we have in the skin which can signal all these different facets that we have in touch? Well, first we can divide the skin into whether it's hairy skin here or the glabrous skin. So the hairy skin is basically the majority of the skin that you have on your body. You have the visible hairs, But just because you don't necessarily see the hairs, like for example here, it doesn't mean that it's not hairy skin. It's still classed as hairy skin. There are tiny, tiny little hairs there. And when we also have the glabrous skin, which has no hairs. So for example, it's the skin on my hand here of the palm. And there is a distinction here because in the glabrous skin here, you have hundreds of thousands of mechanoreceptors. You have so many. It's very, very dense in the glabrous skin. However, it's a lot less dense in the hairy skin. And we also have these, what I said was the C-tactile mechanoreceptors, which are signaling more pleasant touch. There are a lot more of them in the hairy skin here. So we're already starting to think that with the skin here, the glabrous skin, this is for really exploring our world, such as our hands. And then this is more for receiving touch. And then it can be more of an effective skin area here. So when we talk about discriminative touch, like I said, it's more to do with your hands, your feet. You also use your feet and, for example, your lips as well. But we very much explore our world with our hands. And in our hand, we have about 17,000 mechanoreceptors. However, you can actually feel the input from one single mechanoreceptor. This can count. So it's kind of important to think that you have many, many mechanoreceptors and it's giving you plenty of information, but it also is that every single one counts here. It's an amazing system. And the mechanoreceptors are most dense at your fingertip, which is what you would expect, because this is how you're really kind of going out and exploring textures, things like this. It's very, very sensitive to things like this here. And as humans, we use a much more sensory language for touch using our hands. So sensory, what I mean is we talk and we say, for example, something is hard or rough or smooth or cold. So it's less kind of, we say about the pleasantness or the unpleasantness, we do use a more sensory language when we explore the world with our hands. So it's of interest to investigate discriminative touch, of course, such as how we sense the shape of things, the size of things, also the texture and stickiness. And it's kind of a project of mine to actually uncover how you sense wetness, especially with your hands, because we have no wetness receptor, a hygroreceptor. Some insects have this, but we don't have this. Mammals don't have hygroreceptors. So how do we sense wetness? Well, it's going to be mechanical signals and thermal signals, but this obviously arrives very quickly in your brain. And I think it's quite amazing that we have no specific receptor for this, yet we know very, very quickly when something is wet. So keep watching this space for more information on that. However, emotional touch is very much perceived by the hands as well. You know, like I said, when you pick up something and it's maybe dirty or unpleasant, you know very quickly. So you do use your hands still for this effective touch. And so when we look at the density of the receptors over the body, like I said, there are so many in the hands here. There's also a lot on the head. But here is a representation of the human body, the density of mechanoreceptors. And you can see for the rest of the body, it's a lot less dense. However, it doesn't mean that you're not sensitive to touch. And so here is a kind of a very classic map of the body here by Weinstein in 1968. And it shows the difference between pressure and discrimination in touch. So actually the whole body itself is very, very sensitive to pressure. So when I know when something has touched me, but there are more specific parts of the body here, for example, the hands and the face, which are much, much more sensitive to actually discriminating between 2 points. So this is what we call 2-point discrimination. And I like the analogy of, is that, for example, if a fly lands on your back, you can feel that the fly is there. However, you don't know how many legs it's got. Whereas if it lands on your hand, you've got probably a better idea of exactly the size of it, maybe how many legs it's got. So that's the difference between the sensitivity side and the discrimination and how we use our body. One point to make is that the skin changes with age. It changes in thickness. You lose your mechanoreceptors as well. So you have about a 5% decrease in mechanoreceptors for every 10 years. So you actually lose a lot of mechanoreceptors throughout your life. And you can see here, this is kind of the same map with the sensitivity here. And you can see in as the bars get darker, lighter, it's as an increase in age. And here is the difference, the decline basically. And there is some decline across the body, but it's very interesting that actually touch declines a lot. So it declines a lot more on the lower limb, more on the feet, things like this. And this is why maybe older people should be very, very careful, because if they're losing sensation from their feet and they're walking, they may be more likely to have a fall. So all these things, including the mechanoreceptors, are interesting to think about as we grow older and our sense of touch is actually decreasing. It may increase accidents, things like this. Also, the temperature can change over the lifetime. So you're very, very sensitive to temperature, but you're much more sensitive to cold. I think it's probably because cold is a more relevant signal, because then we have some warmth, which is higher than the skin temperature, and then it will quickly go into pain. So for us, cold is actually, you have a much lower threshold for cold over your body than warm. So this is actually quite big. And there's a distinction again between the upper half and the lower half of your body with temperature. You seem to feel temperature much better on the upper half of your body than the lower half. So for example, when you go out in winter, you'll feel cold on the top, but you may not necessarily feel cold on your legs. And this is why, because you have a much higher threshold for sensing cold on your legs. And again, this threshold will increase with age. And so you have to be careful with age, touching different temperature things, because you're less likely to feel the change in temperature and you may get skin damage. So these are kind of things that's important to think about as we age and aging healthily. So I want to just kind of move on to affective touch as well. That was more the discriminative and the temperature side. But our hairy skin, as I said, it contains neurons that we have which are more suited to convey affective touch. And this is what we think is the C-tactile afferents, which is kind of a special type of mechanoreceptor that can convey very gentle touch. And what we think is it actually encodes the positive affective sides of touch, such as pleasantness. And so humans use a much more emotional language for touch on the hairy skin of the body. People will tend to use words such as pleasantness or unpleasantness, and they'll talk about arousal. So it's if it kind of wakens them up or calms them down, something like this. And there are plenty of these C-tactile afferents all over the hairy skin of our body. But again, it's more on the upper half of our body that we know of. And we even do have them in the glabrous skin of our hands, although very, very few. So it may kind of give an impression of pleasantness from your hands here, but it's very different to the pleasantness, for example, stroking the arm, where there are so many more of these mechanoreceptors. So I'm going to tell you more today about how pleasant touch is encoded then with these C-tactile afferents, because what I do is I record directly from them in humans. So I use a technique called microneurography, which is a special technique because there's not many labs in the world that actually use microneurography. It's quite challenging because we put an electrode into a peripheral nerve of a human, and then we actually go into the nerve, we find a single fiber, and we can then record from this fiber. So this is an example setup of microneurography. We will have someone sitting very comfortably in a chair. And for example, we'll go into a nerve of the arm. We'll put a very small electrode into the nerve. And here's the electrode kind of going in. It's quite a lot bigger than the nerve, actually. But we can, if we're lucky, record from one single fiber, even the smallest of C fibers. And so this is an example of what we might find when we're in the nerve. For example, touching here. And the touch is, it keeps on, it's sustained there. We've got a myelinated afferent, a slowly adapting type 1 afferent. And this one, as it kind of says in the name, adapts slowly to touch. So it fires initially, and then it will keep on firing to touch. And it's a real pleasure to actually do microneurography because we can hear these responses in humans. So when I touch somebody's skin, I can hear that exact response. And so this is just a close-up of the spike here. So it's exactly like electrophysiology, but it's in the kind of the real human there, which means we can actually ask them questions as well when we're doing the experiment. So this is an example of the electrodes going into an arm. Microneurography does not hurt, by the way. We are very, very gentle. We put the electrode slowly into the skin, and it's just by a small pressure that it goes in. And most people don't even feel it. They really don't feel it. We're very, very careful. It can take a long time. And this is because we slowly, slowly advance to get into the nerve. And once we're in the nerve, we can then record the signals. And why does it not hurt micronyography? Well, actually, the end of the electrode it's got a slight tip, but it's slightly blunted. And so it's not actually cutting anything here. It's just kind of making its way towards the nerve. And this is an example of a micronyography electrode. And you can see it's a lot thinner than the acupuncture needle, sorry. And so this is why it's possible, because people can sit there for many, many hours with this electrode in their arm, and then we can stimulate their skin and we record the signals. So we're in the nerve. I'm recording from a single afferent. It's like, I found a C tactile afferent. I'm going to record from it. So what happens when we actually record from C tactile afferents? What are they? They are slowly conducting fibers. So they're C fibers. So that means they actually send their message at about 1 meter per second to the brain. So for example, if I stroke here, the input arrives in the brain about a second afterwards. So it's quite delayed, although you don't consciously feel this touch. It's not like I stroke and you feel a second wave of touch. It's not like that. It's very much subconscious. They respond to very, very low force indentations. So it's the softest of stroke, they will respond to this. They have small receptive fields and they respond very, very well to a gentle, slow moving touch. And this is why we think they're encoding pleasant touch, because they also respond very poorly to rapid events and vibration. They're not responding very well to this at all. And they actually fatigue when you repeatedly stimulate them. So these are quite complex fibers. So we think that CTs are not very well suited for discriminative touch. They're not telling you that much about, for example, the intensity, the timing, or the localization. They're much more seen to be encoding the positive, what we think is pleasant side of touch. So I'm going to show you some studies about why we think this. So what we have here is we have a special robot, the rotary tactile stimulator. And what it is, it's a robot which has a brush attached to the end, and this can gently caress the skin at many different velocities and a very calibrated force. And so what we have here at the top, we have a recording of a C tactile fiber, and this is a stroke at 3 centimeters per second, which is very, very pleasant. And you can see it responds very, very well to this. And what we can do is actually we can test different speeds here. So we've got from 0.1 centimeters per second, which is excruciatingly slow. It's almost stationary, all the way up to 30 centimeters per second, which is quite fast like this. And what we find is the CT afferents respond in the middle to optimal velocities between 1 and 10 centimeters per second here. So you go too slow and it's not optimal. The response, the frequency decreases, and you go too fast and it also decreases. So you get this kind of inverted U-shaped curve here, where these velocities here in the middle, which are more these gentle kind of slow stroking velocities, are optimal. And then we can actually do the experiment and we can ask people how pleasant they find all these stroking velocities. And what we find is for exactly the same, we get a similar U-shaped curve that people find these middle velocities the most pleasant. And so when we actually correlate the firing of these C tactile mechanoreceptors with the psychophysical measurement of pleasantness, we get a nice correlation here. So we find this experiment showed that the CTs very much have code the stroking velocity, but they have this kind of optimal speed here. They have a preferred speed, which seems to link up with pleasant touch. To take this further, I did an experiment, it's in 2014 now, we did the same kind of experiment, but instead of having a brush on the end, what we actually had was a probe which could change temperature. And so what we did, we had similar velocities going from very slow to very fast. And then we had the probe at a skin temperature, which was of 32 degrees, which is in the black. And then we also changed it to have hot, 42 degrees, and cold, and that was 18 degrees. And what we find is, as we repeated the experiment, we find that the middle velocities always give the highest firing frequency in these CTs for pleasantness as well here. But when you actually change the temperature, the CTs are decreasing their firing frequency when it's hot and when it's cold. So what we think is that CTs are actually responding to this kind of neutral temperature here and these optimal velocities, which is almost exactly like skin-to-skin touch, a skin-to-skin caress, which is why we think they're so much implicated in social touch, for example. And you can see again, the CT responses correlate well with the pleasantness ratings. And this is just a comparison. When you record from a hair receptor in the skin, the responses are totally different. And this is why we think the CTs are encoding pleasantness, whereas the other receptors are not. So we have both the speed and the temperature of the touch is encoded. However, with pleasantness perception, there is a lot of individual variability here. And so this is not recording anymore. This is going back to psychophysical experiments, asking people to perceive the pleasantness of these brush strokes over their arm. And what we find here is actually there is a lot of variability in the population. So we do have to be careful linking the responses we find in the nerve with the actual perception, because when we get the average, this is something to note as well, we get the average here with the standard error of the mean bars, SEM bars. It's very different to the actual group data here. And you can see there's much more variability. So this is something always to consider in research. What does your group data look like? Because basically it means that we do have this correlation of pleasantness and CT firing, but it's much more than this, because people are obviously finding pleasantness very, very different to each other. So what do CTs signal? Well, we've also found that CTs are a bit special when it comes to cooling. So I said with cooling and with heating, they actually fire less. However, cooling is a special case because we find that warming decreases all the activities in the CT. It just decreases it. However, cooling actually produces a prolonged activity at low frequency. And we still don't know what this actually means perceptually, because what we have here, for example, we've got the warm at the bottom. You can see there are not many spikes. We've got the neutral, and there's plenty of spikes. And we've got the cool, and there's actually lots of spikes here, but they're at a lower frequency. So there's lots of spikes here, the number of spikes, but the frequency is still a bit lower for cool. And so what's actually going on with the cooling? Because when we have this going on here, the people don't actually say that they feel anything different. So this is a subconscious signal, and it may be signaling more comfort rather than pleasantness. So comfort, how comfortable are you in your skin? And this may be due to the contraction of the skin. So you can also look at pleasantness over the body using this special robot by brushing over many different body areas. And I just want to highlight that actually you tend to feel pleasantness very similar over the body. So you have, we tested on the forehead, the forearm, the thigh, the shin, and also the palm. And we get this similar kind of shaped curve all over the body. So it just seems to be that you do have this preference for these kind of middle stroking velocities, but you feel pleasantness very, very well all over your body. So it's not necessarily a single type of mechanoreceptor. It's how you actually feel in your brain can be very different, the interpretation of this. And there's actually no correlation between the sensitivity and the pleasantness as well that you have. So for example, I said you have very sensitive hands in the face, but it doesn't mean that the pleasantness is increased. It seems to be similar all over the body. And I just want to highlight that actually the face is a very special case here. We spontaneously touch our face about 800 times a day, which is really quite big. And self-touch here of the face can also increase with arousal, emotional, or cognitive load. So for example, when you're stressed, you tend to touch your face more. And this is kind of like, this can be a sign of being stressed. But maybe it's also a sign of trying to kind of calm yourself down. So self-touch is an interesting case because we all know that you can't tickle yourself. There's a very well-known study from Blakemore saying that you cannot tickle yourself because you cancel out your own signal here. However, it's more than that. It's a bit more complicated because you can actually, as well as cancelling the signal, you can increase your signal if you want to attend to the touch. So you may tickle yourself and cancel that. However, when I'm actually wanting to kind of go and explore things, I can actually increase that signal, which is I showed in a study in 2012, that it depends on the situation. If you want to feel something, then that signal will increase. And it's also interesting to note that emotions are very, very kind of central here. And the touch from another is usually felt as more positive and more arousing than self-touch as well. And this again is a very big social aspect. There are differences between the pleasantness perception between males and females, and it also changes with age. And I did mention before about touch changing with age, your discriminative touch and your sensitivity to touch. However, for affective touch, it seems to be more complicated. So for gender differences between males and females, there is some evidence that females tend to rate the pleasantness of touch, as higher generally than males. It's a small difference, but it's a significant difference that females tend to rate this higher. We don't know why. It's just one of those things, but it's something to bear in mind. And also age is a very, very interesting thing. We find that as touch sensitivity decreases with age, touch pleasantness actually increases with age. And again, we don't know why this is, because the number of afferents that you have, the neurons, are decreasing, and the C-tactile receptors, we think, will be decreasing. However, the older you get, the more you appreciate affective touch, the pleasantness of touch. So we had a study in 2016 where we had younger people, adolescents, up to 18. We had early adulthood here. And then in the lighter color, it was later adulthood. And what we find is with the same stroking velocities going from slow to fast, we get these nice kind of curves again, where there's a peak in the middle. But what we find is there is just a general increase of the pleasantness with age. And you can see the correlation here. The pleasantness of touch increases. And I've actually repeated this independently very recently. We find the same thing, that the older you get, the more pleasant touch is. We did wonder if it was just a general appreciation of everything as you get older. You appreciate this kind of the sensory input, but we don't think it's that, because in the study, we actually compared the pleasant touch to olfaction. So people were smelling different odors. And for example, a pleasant odor, there was no difference with the age. So it seems to be more of a specific case with touch, that is when you get older, you actually find it more pleasant. And of course, touch has very, very many multisensory interactions. So I just want to highlight a few of the ways that touch can be influenced as well. So for example, touch and temperature. I did say that, for example, these CT afferents, they are mechanoreceptors. They primarily encode touch. However, they do have this temperature sensitivity. And it's true that touch can become unpleasant when it's too hot or too cold. However, that's not always true, because if it's a very hot day outside, you appreciate a colder touch. And again, if it's a cold day, you would like a warm kind of hug. So it depends on the situation and the environmental conditions. Touch can be very much influenced by audition, and it's the kind of the classic parchment illusion. And there's some really good videos on YouTube about this, and if you wanted to see this afterwards. But there's a classic study here. They enhanced the high frequency touch feedback. So they had someone listening to their own touch of a material. And so it's kind of normal shh, 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 like this. And then they actually increased the high frequency. So it went shh, 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 like this. And people actually felt the substance is drier. So you can change the way you actually feel something just by the signals you have when you're actually hearing it as well, which is very, very true. When you're touching more kind of rough items, you hear it. You can hear that roughness as well. The smell as well. So we also have olfaction, and there was a study as showing that unpleasant smells actually can decrease touch pleasantness, as we would all imagine, because, for example, if you're touching something which does not smell nice, it may feel nice, but you want to touch it less. It's quite obvious there. And also vision very much can influence touch. And so there's a study here showing that touching different materials rather than seeing them can actually increase the pleasantness and arousal. So sometimes vision can help, but it can actually decrease the touch signal. Whereas we actually find when something we don't see it, we can actually find it more arousing and more pleasant if it's a nice material. And there's also, I just want to mention that that's cognitive and emotional factors are very, very important in touch. And so it depends on the situation. It depends who is touching you. The signals may be exactly the same on the skin. However, the way you interpret them in your brain can really, really make a difference to an individual to make it either pleasant or unpleasant. So in essence, the signal can be pleasant here. However, when it comes to the brain, it can be a lot less pleasant. So this is why it's complicated to investigate. I just want to kind of finish off by highlighting that a lack of social touch can be not good as well. And so obviously in the time of COVID, we have a lot less social touch and it can be detrimental. And I actually performed this study in 2019. This was done just before the pandemic. And we actually looked at people who had kind of normal touch, who had normal social touch. They had handshakes, they touched people throughout their daily lives. And we had a group who had a lot less social touch. And what we found is that when we tested them on this kind of stroking velocity paradigm from slow to fast stroking, these people here, it's kind of in the black line here, this group of low touch exposure people actually had a decrease in their kind of sensitivity for pleasant touch. They didn't really discriminate the pleasantness anymore compared to the control group, which had this kind of normal kind of U-shaped curve. And so what we find is if you have less social touch, you actually appreciate it less. And so the cycle can continue and it can become very isolating if you don't do this. So I would say it is very good to have social contact and interpersonal touch. And if you lack it, you can come into this cycle of having a lower appreciation of touch, which is, it's not good because it's nice to receive pleasant touch. So overall, just to conclude, we have a need for touch. We all need to be touched. We need it, both the discriminative and effective aspects of touch. So discriminative, when I say that, is more the conscious aspects, the what, where, when. But we've also got this effective touch side, the pleasantness, but also unpleasantness as well. And it can be painful, but it's, to be honest, it's a lot nicer to work on these kind of gentle, pleasant aspects of touch. So we need to take them both into consideration when we actually investigate touch. And there is this high need for touch in society, which is difficult these days. It is difficult. And to really kind of get an appreciation of this social touch, we think we need to activate these C tactile afferents, for example, here, or kind of a pat on the back. And it actually helps with interpersonal bonding. This kind of slow signal coming in can reinforce these gentle tactile interactions. However, we need to be aware that touch can be different over the body. And there are these differences between self-touch and the touch for another, where the touch for another is generally thought of as more pleasant. We found that touch sensitivity clearly decreases with age. However, pleasantness increases. We still don't know why, but you very much appreciate this pleasant touch as you get older. So effective touch is very important, both in self-care, for well-being, and for your social communication. I'd just like to thank my funders and all of you. Merci.
Thank you very much, Rochelle. It was super interesting. And so now we open to questions. So I see a question there.
Okay, hello. Thanks a lot for your talk. Very beautiful. I have a very naive question. Maybe you mentioned it, but I didn't get it. How can we quantify the pleasure? Because there is a scale with numbers, and there is differences, and I think that you use a scale.
Yeah, sorry.
But,
yeah, so basically we just get people to rate how pleasant it is. We have there's different ways of doing it though. So we typically have a scale from very unpleasant to pleasant or unpleasant to pleasant, and they rate it. But you can also give numbers. So I'll say to someone, how pleasant was this touch? And they say 10. And then then they do kind of a fast one. How pleasant was that touch? Well, it was a 2. So there are different ways of doing it, but they're kind of equivalent. You can use all these different ways. But here it was unpleasant to pleasant.
Thank you. Thank you.
Thank you very much for your talk. I had a question about the gender difference. You said so there is a difference between male and female, and I was wondering if you also checked with the odor, like as you've done for the holders, and if it could be related to the expression, like we know in sociology that it's less that the male did not show and express emotion the same way as women. So would it be like in the expression, or would it be also biological? And do you see a difference in the CT activation, for example?
No, it's a really good question. To first answer about the CT activation, I haven't seen any differences between males and females, but that's not to say that there are differences because there's only a small number of these C-tactile afferents that have been recorded from. However, I don't think there are differences in the mechanoreceptor physiology between males and females, as in the mechanics, the firing. However, it can get a bit more complicated than that because it's various different levels where there could be these differences. So I've talked with other kind of team members, why potentially do females experience pleasantness slightly higher? It's only slightly, so it's not all studies, but in a general meta-analysis, they did find that females do rate pleasantness on the whole a little bit more higher than males. It could be a number of reasons. It could be hormones, things like that, because hormones can actually change the state of the skin. They can make it softer, so it may be more pleasant. Also, females, so basically humans, will probably have around about the same number of mechanoreceptors per human. However, males then are typically bigger than females, and so their density is slightly less. And so for females, they tend to have more sensitive skin because the mechanoreceptors, it's the same number, but they're slightly more dense. And so it could be the same for CT afferents. So you may, with the same brushstroke, actually be activating more CT afferents. Whether this actually counts, I'm not sure, because it could be more, again, like you say, it could be more of a social level, emotional level as well. The females tend to express themselves more through touch. And so it's potentially that as well, or all of these factors. It's difficult to kind of pinpoint one exactly, but it could be a number of levels. It's complicated.
Hi. Thank you for your talk. The first, my question is about the difference between like how can we differentiate between self-touch and other touch? Because I get it, but I don't understand it because I can actually feel when I touch myself, and sometimes like it's calming, etc. But I understand that, for example, with the tickling. So do you know, like, is it known if there might be like high cognitive mechanisms of self and others involved?
Yeah, no, it's a very good question. It's a complicated issue. The difference, what is the difference essentially between self-touch and other touch? So when you have self-touch, you have a motivation. You yourself choose to go and touch your own body. And so not only do you have the kind of the sensation coming in from the skin that is touched, which will be other touch, you have the intention of movement, you also have kind of the activation of your muscles, you have the feedback from your muscles, and you have the feedback from the skin touching. So it's already a complicated system. However, we think that there are certain areas in the brain which will cancel out this signal, such as it's more the cerebellum, which will do this kind of self-cancellation of the signal. So you have the descending signal saying that this is the movement, and then you have the touch itself. And in the cerebellum, it will compare the output with the input, and you can actually subtract it away. And you should be able to kind of then know when you're touching yourself. However, I would say it's more complicated than that. The case of self-touch, when we measure it experimentally, you go in and you ask people to kind of touch their arm or touch their face or try and tickle themselves, something like this. It's quite dry. And so there is lots of examples of self-touch in everyday life where self-touch can be very calming. For example, kind of touching your own face, applying something to your face, or grooming. It's more the case of grooming. It's important. So as mammals, we do have a need for grooming, and you see kind of the videos of monkeys grooming each other, things like that, but we don't tend to do that as humans anymore. We self-groom. So I think the case of self-touch is very, very important, but for us to look at, it's complicated. It really is complicated. And so there are some very... Is there a motivation for self-touch, such as grooming, for example, finding that ingrowing hair or something like this? So yeah, it's I think there's a lot more to explore as well.
Sorry, were you at the question? I think. Okay, so wait 1 second. First, it was let's go by order because so we don't.
Thank you a lot for your talk. I was wondering, it's a bit a silly thing. If you will ask the people when you took different age groups, like adolescents, young adult and old people, could the perception of the body also influence how much you feel the pleasant from the self-touch and from the touch of the other? Because I could imagine that for adolescents, they have very hard relationship with their body itself. So maybe this age difference, it's coming from the self-acceptance of the body. If I don't know, it's just my guess. And also, could it be for the adult people as a lack of social communication, as in a kind of social isolation? And if you ask people to evaluate their relationship with the body, could you think it could explain this age difference? Thank you.
No, these are the exact factors that we need to think about. And these are the things that need exploring. There isn't much work out there on pleasant touch as we age. There are very, very few studies. And it's something that we need to look at more. Why? Why does it increase? And so I think you bring up very, very good points here that, for example, when you're young, your body is changing, you're growing, your self-perception is changing. And so it's not necessarily a pleasant process either. And then you go into kind of adulthood, you've probably got a lot of social touch in your life. And then, for example, when you have children as well, you have a lot of social touch there. And then it's true that when you age, you probably have less social contact. You may retire, you have less contact with people at work. And so the social side of your touch will decrease. And maybe that's why people feel it as kind of more pleasant. It's true, as an appreciation of touch, although the body very much does change throughout the lifetime. And so I said your mechanoreceptors do decrease. And so it may be the case of touch feels less sensitive when you're older. And so you appreciate it because you are still feeling something. So you may still have these C-tactile mechanoreceptors. They may be preserved, we don't know. But no, it's definitely a point to actually investigate. How does affective touch change throughout the lifetime, and why? How does that kind of relate to the body image as well? Because I think you grow older, your body will change and your skin becomes different. And so people, when they're older as well, may be less content with their body, the kind of natural aging process. And however, they still feel it's pleasant. So that's kind of maybe in conflict a bit there. So I think there's very, very many different levels to consider here, but that's something we want to definitely do in the future.
Thanks a lot for the talk. It was very interesting. I had the question about, you showed that the adolescents, adulthood and late adults. What about babies? We know that the relation mother-baby, it's very important, skin to skin, and a lot of affecting touch. I know it's a little bit difficult to study on babies, but there was any studies about that or not at all?
No, there are a handful of studies looking at touch in very, very young children, babies. However, you can't ask them. This is the problem. You can't ask them how pleasant it is. However, you can measure physiological variables. They have done kind of various different studies looking at heartbeat, how it decreases, things like that when you touch, and they get the same effects. When they receive pleasant touch, what we think is that the system is there, it's active. These C-tactile afferents are there from birth, and they are very much active. In fact, it's likely that because when you grow anyway, you do lose neurons, but it's very likely that you have many more CTs when you're younger, and they are there to convey this effective touch. So there is, yeah, as I said, kind of some research on babies, and it's shown that they do have the same reactions as us. So we do think that they are receiving the pleasantness. We did have, to the study that I showed here, we did have a separate paper which actually investigated touch in younger children. And that we use smiley faces rather than a rating scale because it's difficult for them to actually say. And we did find that they have a similar touch appreciation, that they preferred the kind of the middle stroking velocities as well. Although we couldn't necessarily say it's any different to adults, they're showing the same thing there.
Hi, thank you for the talk. It was very nice. I had a question for people who have like important lesions like severe burns or stuff like this, and they have to undergo like a skin transplant and so on, and they have like very big areas of their body that just go numb and they don't feel anything. Do you know, or have you seen if they have like some kind of consequence of this lack of sensitivity in their body, like they feel this lack of touch, and if they just lose their, like you said yesterday, that the less touch we receive, the less prone we are to look for this kind of effective touch. Like, have you seen anything like this in people who have this kind of like lesions?
It's a very good question about, if you have damage to your skin, how does it change your touch? Obviously, it changes the discriminative side of touch. You feel things a lot less, but it's going to change the effective side as well. I don't think there are many studies on this. I can't think of, but it's a difficult case to study as well, because when you touch over a burn area, it can still be painful. You can still have receptors there, and it's more the nociceptors, which now have been sensitized. So you have different nociceptors for pain, and it's quite likely in a burns kind of patient, for example, that there is a higher sensitivity here to pain. And so we don't necessarily want to touch skin which has been damaged. And this is something we do in our studies. We say we don't want to recruit people who've got any skin damage, because we don't want to hurt them as well. However, there are kind of models of touch, and so you can actually induce allodynia, for example, so an increase in sensitivity. So you can have kind of a small kind of experimental burn, something like that, by applying capsaicin, and then you can touch near it as well. And we do find that there are differences here, and people do think that the CTs may actually be kind of signaling the kind of like the more pleasant side of touch here. So for example, when you fall over and hurt yourself and you rub it better, you're kind of activating these mechanoreceptors, not just the CTs, but the other ones. And this increase in tactile feedback can decrease the level of pain. However, not always. And so if something's very sensitive, it will just always be painful, unfortunately.
Yeah, I also have a question. Sorry. I use the opportunity. Yeah, very fascinating topic. I was wondering, you compared a group with low touch and a group with normal touch. And I was wondering how you recruited the low touch group, and if you controlled for covariates, like, for example, psychopathology or recent life events.
Yeah, no, we did kind of like it was a bigger sample, and then in the sample we had a look at all these, because we measured different kind of cognitive things, and so we didn't want to recruit people who had a life event like that. These were really people who just experienced a lot less touch in their lives, but it could have been the situation, for example, more of those people lived alone. Things like that. But we just wanted to recruit from the general population. And then we selected people from a control group, which was just kind of the normal values that we found. And then we had people who, we had different scales that we used. So I think it was a need for touch scale, something like this. We had different ones. And from that, we did actually identify a group of people who responded less, who just had a lot less touch in their lives. So it was very much selected from the general population there.
Thanks. Actually, I put back that. I noticed that in the action propagation speed is in the range of the stimulus that you use. Mm-hmm. And I was wondering whether there was any effect of synchronization of the afferents towards an integrator at the spinal cord or not. That's, for instance, if you are exactly at the same speed as the propagation, all the action potential may arrive at the same moment in the integrator. Mm-hmm. And one way of testing it would be to use stimulus direction, which is parallel to the fibers or perpendicular. Is there any effect that is known? Because it's also, if I don't mistake, on the range for the highest pleasantness, no?
So you're saying about if I change the direction?
Yes.
Like that. We have changed the direction.
Does it affect anything?
No,
not at all.
No. There is a receptive field, which it can be a bit variable. So in the whole, I would say you change the direction and it doesn't change the response. However, not necessarily in all units, because you can, with the CTs here, they have like, actually the receptive field is very small dots, and it can I think it's between 1 and 8. And so it depends on the receptive field. They're really quite complicated, and there's a lot more that we need to look at. And it's true that I think if that this receptive field is stimulated in a different way, we may be able to get a different response. However, when we take into account a piece of skin as a whole, I think the effect is kind of cancelled out. But on a single receptor level, it's a bit complicated. And it's possible, for example, that we may be able to fatigue one area of the receptive field and you get a slightly different response. There's things that we want to look at when we can get back in the lab and do experiments, for sure.
So we have other 2 questions here.
Hi, Rochelle. I have a question about the difference between touch and pain. So we had this question about burn, and it made me think about this. And then, so touch is among the classic senses. Pain is well known. And now you have this pleasant touch. So how different actually is touch, pleasant touch, and pain?
That's difficult. That's difficult because actually we have pleasant touch, which is very gentle touch. But that very, very gentle touch, even for example, a fly crawling over the skin, isn't always pleasant. When it's a very kind of too gentle, that's not always a pleasant touch. So I would say pleasantness is in the head. The signals coming in are low threshold mechanoreceptors, and the pleasantness is very much a perception. And so for me, when I actually look at the skin, you have a range of mechanoreceptors. And so for these C tactiles, they will encode very, very gentle touch. But the other A beta mechanoreceptors encode the same forces. The CTs are not special in the threshold for the level of touch. There are other ones which are even more sensitive to touch. It's just that we don't find the same relationship with the pleasantness. However, it's interesting because then when you go to more harder touch, we've not actually explored how the CTs fire to more forceful touch, and they will fire. But then you'll have the nociceptors, what we think is the classical nociceptors, but they're not really nociceptors, because a touch which is like this will activate nociceptors, but that's not painful. So pain is in the brain as well. It's kind of the nociceptive signal here is not necessarily pain at all. And so in fact, you've got these C nociceptors, which are rather low threshold. And sometimes some will be activated if you gently stroke with a brush over the skin. That's how kind of low threshold they are. I don't think they're even necessarily signaling pain. They're just signaling more forceful touch into that region. But then again, you have the real nociceptors, which are signaling higher mechanical thresholds, for example, a cut to the skin, something like that. And that's pain. So yeah, the skin is really quite complicated, and the different signals between the periphery can be very different when it actually arrives in the brain. But it's very much a perception of pleasantness and pain. Whereas for me, the periphery, the skin, is the kind of nociception or mechanoreception, and it doesn't discriminate so well there.
And I have another one, sorry. For the 2019 study, the latest one I think you presented, could you clarify whether the individuals who were lacking touch, was that during development or later? Because I was wondering, sorry, if you have somebody who lost touch at 20 or 30 years old, can you predict some impairments, like psychologically or in touch?
Yeah, those people that they do have a normal sense of touch. They can feel normally like everyone else. It's just that they had less social contact. There are a few studies now looking into kind of factors like that, especially for more when people are younger and they have less touch, how it impacts on their lives. And if they go through their younger life with a low level of decreased touch, for example, it will impact them. However, it can be remedied. If they go into a situation with normal touch, things like this, they will work out hopefully fine. But yeah, it's this was very much a preliminary study, and we haven't followed it up yet or anything like that. It's difficult to do so, but it's definitely interesting questions to think about for the future.
Thank you a lot. So we're running late. I will take the last two questions. I think, do we have a question online or? Okay, so one question was already booked here, sorry.
Hello, my first question will be, the first time, how could you be CT nerves are new nerves, you know, and not just the usually pathway used for the touch? And my second question will be about anatomy and cartography about CT nerves, and does it go through the same pathways such as sensitive touch in the cortex, etc.?
So when I say that they're more like newly discovered, it's like that, but they've always been there. So is that more your question, whether they're, because they're actually it's an old system. It's a very old system. But we didn't think they existed in humans. We knew that they existed in animals, but they were only found later in humans. But they're very much there. It's very much an old system. Was that the first question?
It was about how could you say it's new in the sense of we did not know this earlier.
It's because people hadn't found them. It was actually the technology. And so microneurography like this has existed for over 50 years. However, the recording, the technology has got a lot better. And these C fibers are difficult to record from. And most people will go into the hand in microneurography. A lot of studies have been done on the hand, a lot less on the hairy skin because it's a lot more difficult to access. And so they were only more recently discovered compared to the other mechanoreceptors in humans because people just hadn't been in these nerves. They hadn't looked for them. And then they found them and they're abundant. They're very much there. And so for the pathway as well, it's been mapped relatively well in animals. We think it will ascend in the spinal cord, but in humans, it's difficult to say because we would expect it from the animal work to ascend in the anterolateral tract with C fibers, things like that. However, there was a recent study showing that maybe that's not the case. We're not actually sure for pleasant touch. So we know that the C fibers, the C tactile fibers will go into the spinal cord, but we're not quite sure exactly where they're going up. We still don't know. And then they'll eventually go up into the brain. And we think that they will activate the insula, but it's not just the insula. There are kind of other areas as well where we think that the CTs go, but it's very difficult to study this pathway, especially in humans. And when we actually get up to the area of the brain as well, the CT input is very much mixed in with all the other touch input coming in. And we're kind of developing our knowledge more and more, but it's very much a question that we need to investigate.
Okay. Okay, so actually, like, how many questions would we still have in the room? Please, okay, raise your hand. Okay, 2. Then we have one question line. So we have a lot of time for the posters, and we thought maybe we can use 10 minutes more now and have 10 minutes less in the posters, okay? So one question online is actually, okay, can you ask, about cultural differences in touch? How does it affect people in these societies?
So there are some cultural differences in touch. However, there are not that many studies out there which have really investigated that. We do have, so for example, the sensitivity to touch, people have the same sensitivity, and that's not really going to change between different humans. However, the appreciation of pleasant touch can be different, and so we don't really find that many differences between our measures of pleasant touch. For example, we have this kind of general stroking curve, and people find pleasantness similar between different cultures. However, there are cultures out there which are a lot more tactile, which will touch a lot more. And so it could be that they do appreciate pleasant touch more. However, there isn't actually a systematic study out there But one good thing about the COVID pandemic was that there has been a lot of online studies now, which have been going on online. So it's across the whole world. And the data is being analyzed now. So I can't say so much about it because it's just about coming out now. But this has been an opportunity. One of the few advantages of the COVID pandemic that it's opened up global research. And so hopefully there will be studies coming out onto how, for example, the pandemic has affected social touch between people and between countries with high levels of social touch and low levels. So that'd be interesting to find out. But at the moment, we don't know.
Thank you a lot. And thank you for answering to all of these questions. I mean, you're between yesterday night and today. Thanks a lot. And thanks
questions are great.
All of you for your
questions. Help me. I really enjoy questions because it's things that help me in my research.
I was just wondering, coming back to this low touch group of people, like we know that there is a lot of plasticity in the body, and actually maybe this plasticity give this low sensitivity. Do we know if there's a way that it comes back when if they found some touch again, or if it's not like if they're doomed?
No, it's a good point, and we haven't followed these people up or anything like that. However, yeah, it's a question is if they are in a situation where we give them more social touch, they have much more social touch in their lives, will they appreciate it? I think for some people who have a need for touch like that, it's just their situation. It will increase their pleasantness of touch. However, there is a lot of individual variability in their appreciation of touch, and some people just don't like social touch, but that's okay. It's okay. We're all different. And so forcing social touch on people who don't like it is not necessarily good either. So I think we'd have to be careful. But I think for those people who want it, but who miss it in their daily lives, if they have it, they should appreciate touch more.
Hello again. Continuing with these questions about people that have like not too much effective touch. We have been talking about how effective touch can modulate certain behaviors, but does it happen in the opposite way? Like people who go into depressive states or that have some kind of distress, do they have like a decrease in the sensitivity of effective touch, or do they experience like they just want less effective touch, or they just feel it less, or have you seen if there's any difference in these people on how they perceive this effective touch?
What kind of people? What?
Like people who have like, I don't know, psychiatric distress, like depression, anxiety, or this kind of disorders.
There are a few studies out there now looking into effective touch in different kind of patient populations, things like that. I would say that it's complicated because even here I presented the individual variability in a group of kind of normal control subjects, and we find a lot of variability in the appreciation of touch. And so when we go into a group of more kind of a patient group, we find that the variability can be even a lot higher. And so there have been some studies, for example, on autistic people and their appreciation of a pleasant touch. And they kind of find some effects, but they're not clear. They're not clear. They can still appreciate pleasant touch, even if they don't particularly necessarily want it even. They don't want the touch. They still appreciate it. However, it tends to be the case is that their responses are more variable, and the variability may be a problem there. There are kind of a special group of patients who they lack some of the C fibers, so they have a general decrease in C fibers. And again, we find that they're kind of this curve that people have, we find that it's a bit lower, but it's not hugely lower. It's only a little bit lower. So they can still appreciate pleasant touch. It's just compared to the general population, it's a little bit lower. But I think that we have to be very careful with our studies on pleasant touch, because just because somebody doesn't have a high appreciation or this kind of nice curve, it doesn't mean that anything's wrong with them, because it's highly variable. And then we go into these different groups and it's even more variable. So it's not like we can ever diagnose a problem through pleasant touch, because it's too variable, I'd say.
I also have a question if no one else has any other questions. Okay. I was wondering what about amputees? So if someone misses a leg or an arm, do they feel the same as normal people feel in the other parts of the body. I mean, control people. Or also, what about the phantom limb? Like, are there studies studying like possibly like affective touch on phantom limbs?
That's it's something that we're actually interested in. We do work with some amputees in the lab, things like this, and we're planning studies on amputees. I don't think their sense of touch particularly changes after an amputation. However, I don't think anyone's really investigated pleasant touch kind of in a phantom, because it tends to be more painful. Phantom limbs tend to be more painful. I don't think people tend to report necessarily pleasantness. They just tend to report that it's either there or it can be a bit painful, or it's not there at all. However, there are some studies looking at the rubber hand illusion, things like this, and how pleasantness can affect this. And I think even there is quite new, but I think it's a little bit inconclusive. That we would think that activating the C fibers more might give a higher sense of embodiment, but it's not necessarily true. But one thing in my future work is that I showed this person, an amputee here with his prosthetic arm, is that we're working with people like this, and we're working on nerve stimulation as well. And so we know that you can actually go into a nerve and you can stimulate single fibers, and you can get a percept such as vibration or pressure, and that can be very clear. However, when you stimulate a CT, you don't get a conscious percept. So one day I would like to be able to provide kind of all different sensations such as wetness, stickiness in amputees with prosthetics. However, giving pleasantness, that's going to be a difficult one, because it's also a very subconscious signal. However, if we can hopefully stimulate remaining fibers, these C fibers, you could have maybe temperature, pain, and maybe pleasure as well. But that'd be a very difficult question to answer, but it's an important one. Because if you really want an operational prosthetic arm, for example, you need to be able to not just feel the general kind of normal sensations of duration and pressure and things like that. You need to be able to feel temperature and pleasantness, things like that, the whole thing. Maybe even better than us. We'll see.
Thanks a lot. So if no one else has any other questions, I will thank you again for this amazing talk. And I think discussion, like you can see how amazing it was by all the questions that were asked.
Thank you for all the questions. They're really, really good.
So now we have the second poster session. So everyone actually is free to stay at their poster, go around, do whatever you want. And then afterwards, in 1 hour, I think 45 minutes, we will have the award ceremony for the best posters. Woo! So yeah, see you later. No.
Use the QR codes around the poster session to sign up for the event today.